Okay. Last time, what did we talk about? Vision, eyeballs to, yeah, so hemispheres. So my right hemisphere is receiving what sort of visual information? The left eyeball? Left side of space, right? So the visual input is upside down and backwards on your eyeballs, right? On your retina. And then the retinas either send all the information to the same side or the opposite side, depending on whether they're the temporal hemiretinas, so on the outside, or the nasal hemiretinas on the inside, next to your nose. So nasal hemiretinas cross over, temporal hemiretinas stay ipsilateral, right? So contralateral versus ipsilateral. Okay, and then we got up to the LGN, to the lateral geniculate nucleus. So at the level of the LGN, what do those cells know about the visual stimulus? What do LGN neurons know? Like, what sort of information are they getting? So they're the sensory switchboard, right, at the thalamus. But what does that mean? What sort of input are they getting? Do they know anything about color? Not really. Do they know what the shapes are out there in the world? Not really. So it's very low-level information that they're getting. So what they're getting is basically, really what they're getting is an increase in the firing rate of the retinal ganglion cells, or not, right? And the retinal ganglion cells increase or decrease their firing rate depending on whether there's light present on that part of the retina. Right? So if light comes in, strikes a certain part of the retina, that signal gets sent to the LGN. This picture, I love this picture. This picture is beautiful, I think. It reminds me that the brain is not color-coded, first off. It's not usually black and white, it's usually pink, right? So you have the optic nerves coming in here, optic chiasm. This is where that crossing over comes in. This is the optic tract here. And then this is the lateral geniculate nucleus. Geniculate means knee-shaped or bent, and so you can see that bend right here. Isn't that cool? And then from the LGN, you get these, they're called optic radiations that come out here and sort of swoop down here to the primary visual cortex. I love that orderliness of the whole system. It's gorgeous. Okay, it looks like what? Like a muscle, yeah. Yeah, like the muscle fibers, how they're all sort of strung out like that. Yeah, pretty cool. All right. Yes?
Were the tubes blood pathways? Like
Sorry, which tubes?
I don't,
like these right here? Yeah, those are blood vessels right there.
What about those right above? It's like a tract.
These here? Anybody know what that is?
I should know. Those are the olfactory.
That's the olfactory bulb, yeah. Yeah, so those are your olfactory bulbs that have the little olfactory sensory neurons that poke down into your nasal cavities. So that sits right above your nasal cavities. Yeah. This right here is the LGN. Yeah, so the knee-shaped structure. So, yeah. Cool. All right. Any other questions about neuroanatomy? Okay, so if we talk about really any sensory neuron, specifically a visual sensory neuron, we have to talk about their receptive field. So a receptive field is defined as the area on the receptor surface that when stimulated influences the firing rate of that neuron. So it's a property of the sensory neuron. Think about it as the area on the retina, which corresponds to an area out there in space in the visual field, right? So if there's a stimulus in that part of the visual field, this retinal ganglion cell will increase its firing rate, right? So you'll get more action potentials if a photon of light comes from that area, from that spot in space, and strikes your photoreceptor right there. Does that make sense? Everybody with me so far? So light comes in, hits the retina in a certain location. That signal now goes to the LGN. So the LGN knows that there's light in that specific location. Okay? What else does it know? Not much. Right? So individual LGN neurons only know if there is light in that specific location of the visual field. So consider a pixel. it's like each LGN neurons represent one pixel in the visual field, in the overall picture, right? So if all you know is, oh, hey, there's green here because the certain cones are firing in a way that corresponds to green, then great. Does the LGN therefore know what objects are out there in the world? No. So it just knows that there's green in that specific location. If you combine the activity of several LGN neurons, though, now you get a little bit more of a picture, right? So at the next level, you're going to combine the several receptive fields to create a larger receptive field that's more complicated. Anybody know what this is?
Strawberry.
No. Good guess. Aloe is a better guess. It's a cactus. All right. Did you guess cactus? Okay. So if you put all the pixels together, then you get the full picture, right? But at the level of the LGN, all the LGN neurons know is there light in a very specific place? So if there is a point of light out there in the world, they'll increase their firing rate. So these are called on-center surround receptive fields. So there's an area on the receptor surface, on the retina, that when light strikes it, this LGN neuron will increase its firing rate. If the light comes in slightly to the side of it, it'll actually decrease its firing rate. So you get an excitatory center and an inhibitory surround.
Why is that?
Ooh, great question. Anybody, any guesses, any ideas why that would be? Why would you want excitation in the middle and inhibition on the outside?
Does it make the image crisper?
Yes, it does.
Makes sense.
Yeah. So it enhances what's going on in the middle by creating some contrast. Okay, so these are center surround receptive fields. Excitatory and inhibitory effects are found in the receptive field. So you can have excitatory in the middle, inhibitory on the surround. You can have the reverse as well. So some neurons will be inhibited in the middle, excited on the outside. Yeah, excitatory center. Yeah, exactly what I just said. Okay, so these small receptive fields of these retinal ganglion cells will all converge on a single LGN neuron. And then you can have several LGN neurons, each of which that have round receptive fields will all converge on a single primary visual cortex neuron, or V1 neuron. And then V1 neurons like lines and edges. So how do you get lines and edges from single round points of light? Add them all together, right, yeah. So stack them all up in a line, and you can get a line or an edge. So sort of like this. So if these are the receptive fields of a bunch of LGN neurons, and they all synapse on a single V1 neuron, if they're all excited by a single bar of light, for example, then this V1 neuron will fire strongly, according to the graphic here. Yes, Maria.
Again, the contrast between like why it's like positive on the inside and negative on the outside, and then vice versa. Like I'm just trying to understand when and why.
Okay. The when is kind of random. So some neurons have excitatory centers, some have inhibitory centers. Why, like a particular neuron has that set up, we don't know. It's fairly random. Most of them are going to be excitatory, though.
But will it produce a different effect? Is what I'm kind of wondering.
Yes, they will. So they'll, at the level of like the V1, you get fairly complicated responses at V1. So this is a very simple cell. And in fact, that's what it's called, is a simple cell, a simple neuron. There are complex neurons as well that respond not just to lines of a certain orientation, but also to lines of a certain orientation that are moving in a certain direction. So in order to get those more complicated responses, you need more complicated inputs, basically. So that's the why for why it is, why they like end up being like that, like what designates one being excitatory and one being inhibitory is kind of random.
Oh.
Yeah, does that answer your question?
Yeah, I just didn't know if when it was either or, because it has to be one or the other.
Mhm.
I just didn't know if it was going to produce a different effect of what you're
Yeah, so it'll help you with the different things that you can see, right? Because we don't just see lines and edges out there in the world, right? We detect motion, we can see color. Like there are more complicated things, and the inhibitory and the excitatory help build up that more complicated representation. Okay, cool. All right, how do we know this? Mean experiments with cats. The cats are anesthetized. They are humane experiments with cats.
The researchers actually listened in to individual nerve cells firing in the anesthetized cat as they presented it with different visual images.
When we started working, Torsten and I, in the late 50s, we set up our first experiments, and they didn't go well because at the beginning we couldn't make the cells fire at all. We'd shine lights all over the screen, and nothing seemed to work. And rather by accident, one day, we were shining small spots, either white spots or black spots, onto the screen. And we found that the black dot seemed to be working in a way that at first we couldn't understand until we found that it was the process of slipping the piece of glass into the projector, which swept a line, a very faint, precise, narrow line across the retina. And every time we did that, we'd get a response.
All right, so those ticks that you hear in the background, what were those? Neurons firing, yeah, action potentials. And so this particular V1 neuron that they're recording from in the cat likes a line of that orientation moving in that direction, right? Which is kind of very, very basic, right? Very, very simple. So these guys, David Hubel and Torsten Wiesel, they actually won the Nobel Prize for their work on the primary visual cortex of cats. And discovering these simple cells, because at first what they were showing the cat was, pictures of mice, right? Because cats like mice, pictures of circles and squares and blah blah blah, all of these like fairly complicated shapes. But it turns out that the cells in primary visual cortex don't care about complicated shapes. It's much more basic than that, right? So all that they care about are lines of a certain orientation in a certain location. Oh, we got this to move. There we go. Right, and the way that you get that from your round receptive fields in the LGN is that you have them all sort of stacked up on top of one another. Have them all line up, synapsing on a V1 neuron. If you have a line of light that's just off your preferred orientation, you'll still get some excitation. You'll also get some inhibition, and so your firing rate it will go up, just not as much as for your preferred orientation. So you get what's called a tuning curve around that preferred orientation. Yeah?
So it goes from the ganglion cells to which cells, then to the V1?
Retinal ganglion to LGN to V1. Right? So retinal ganglion cells are in the eyeball, LGN is in the thalamus, and then we're into V1. So V1 neurons have now rectangular receptive fields. So rather than round receptive fields, we can stack them all up and create a rectangle. Yeah?
In the previous diagram, you didn't have the LG represented. It was just the first and the last stage. Is that what that was?
Yeah, so it's sort of skipping a step here.
Okay, so that's
Sorry, yeah. So this should be, there's an additional step in here.
Thank you.
Yep.
On the last slide, though, the one before, though, right?
Yeah, let's see. This one, yeah. So here you've got all three steps. Here, for simplification, it just has the two. Sorry, I didn't realize that. Okay, why lines and edges?
It helps you determine where things are.
Right, exactly. It's going to help you determine where things are in space. And so let's say that I want to pick up the water bottle. What is the important information about the water bottle? The cool stickers I have on it? No, it's the lines and edges that correspond to the outside outline of the water bottle. If I want to walk through that door, what's the important information? Where the edges of the doorway are, right? So I want to know where those lines are because they correspond to the edges of the object. So interacting with objects out there in the world is enhanced and facilitated by lines and edges. Also, lines and edges, so this is a really old picture actually, I'm realizing now. Have you guys seen Lost? Yeah, okay, it was popular. It was a popular television show in the early 2000s. Anyhow, these people are ridiculously good looking for people who are stranded on a deserted island.
Is she pregnant?
Yes, she is, actually. Yeah, so that's a major plot point, is that she has baby on the island. But if you want to know where Michael starts and Jack stops, what's the important relevant information? It's going to be the lines and edges, right? So if you can decompose the scene into lines and edges, and this is just a really simple Photoshop filter that I passed over this, that just enhances the edges, you can figure out where Jack stops and where Michael starts, right? You may notice in my quizzes that Jack and Kate and Sawyer actually feature kind of prominently. I was a fan at one point. Okay, also, edges are invariant to lighting conditions. What does that mean? If I turn the lights on in here, what's the total amount of photons that are going to be hitting your retina? It's going to go up, right? So right now it's pretty low. If I turn it up, or if I turn on the lights, or if you go out into bright sunlight, it's going to go up dramatically. But does that matter for lines and edges? No. So lines and edges are invariant to lighting conditions. So if we change the color of the light, if we change the amount of light, doesn't matter. The lines and edges are going to stay the same. So you can still, interact with the world, walk through the doorway just fine if you go from an area of low illumination to an area of high illumination. Okay, so first thing the visual cortex does is break everything down into its component lines and edges. And the way that V1 is organized is actually really interesting. So if you look at the surface of the cortex, of V1. So let's say V1 surrounds what's called the calcarine sulcus at the back of the occipital lobe. So if you go perpendicular to the surface of the cortex, and if you record from a cell at the top layer, it's going to have a rectangular receptive field. It's going to respond to lines of a certain orientation and mostly respond to input coming from one of your eyes. Yeah?
Just to get this clear, the V1 is the cortex?
Uh-huh. Yeah, primary visual cortex. Let me write this down. Nope, not with that one. Cortex equals V1 equals, other names for it, calcarine sulcus. Equals Brodmann's area 17, striate cortex. No, you don't need to know any of these. You may see them occasionally. You might see striate versus extrastriate cortex. So striate cortex is primary visual cortex. Extra, because if you stain it, it's striped. Extrastriate is the rest of the occipital lobe except for primary visual cortex. So you may see that every once in a while. I'm going to use V1 every time, or primary visual cortex, both of which I think are pretty descriptive of this is the first area that receives visual input, thus V1. Any guesses what primary auditory cortex is called? A1. A1, yes, very good. Primary somatosensory cortex? S1. Primary motor cortex? M1. Very good, yeah. All right, so the neuroanatomists have done us a favor there. So if there's a V1, that implies that there's a V2, right? Which in fact there is. There's a V2, V3, V4A, V5, and then it gets a little more complicated after that. All right, so in V1, if you record within a column, so this is a cortical column. At the most superficial layer, the top layer, you're going to get a receptive field and a preferred orientation and a preferred eye. If you move down to the next layer, same thing. So you're going to have an overlapping receptive field, the same orientation preference, and the same eye dominance. And that's all the way down into the cortex. If you move across the cortex, though, as in this scenario, you'll encounter a certain receptive field, orientation preference, eye dominance. But then as you move over, your preferred orientation is going to change just a little bit until you go through 180 degrees, and then it flips around again, right? So all of the preferred orientations are represented for overlapping receptive fields, but not identical receptive fields. And the eye dominance, you can't really see this yellow on gray here, but the eye dominance changes as well. So you go from right eye dominant to sort of bilateral to left eye dominant. And so you'll change gradually over the surface of the cortex. So if you put it all together, you get what's called a hypercolumn, where within this entire hypercolumn, you'll have cells that respond to 180 degrees of preferred orientations and will respond to input coming from both the left eye and the right eye. It's very orderly. It's very organized. Remember those optic radiations that come from the LGN to the cortex? Part of the reason that they're so organized is to maintain this organization. And so, because it's so organized, you can build up an entire map of what's going on the retina based on what's going on in the V1. It's called a retinotopic map. It's not a very good map, though, in that there's a massive distortion. So if you look at the area of this is the visual field. So the macula is the area that surrounds the fovea. So let's call this the fovea right here. So how big is the fovea? I told you last time. About the area of a quarter held up at arm's length. So fairly small part of the visual field. But if you look at the area that represents the fovea in the cortex, it's about a third of V1 is devoted to representing just the fovea. Why? Why have so much cortex devoted to such a small area of the visual field? Yes?
That's like all our detailed vision. I mean, that takes less processing.
Right, exactly. It's like having a high-resolution image, right? So a high-resolution image on your computer, how much space does that take up on your hard drive versus a low-resolution thumbnail? So the higher the resolution of the image, the more space it takes to represent that. Same sort of thing going on here in the brain. Exact same idea. Your brain is not a computer. Your brain is much better than a computer, but the same principle applies here. You need more neurons to represent that detail vision.
So what does the macula do?
So the macula is just the area around the fovea. So
I've heard of macular degeneration. Is that related
yeah,
to the macula?
it's yeah, exactly. Yeah, so that's when your macula, either the cells are degenerating or they may detach from the back of the eyeball. So remember that point of focus that's supposed to be right on the back of your eyeball, right on the fovea. If your retina has detached from the back of your eyeball, it's going to move relative to that point of focus, and so your vision is going to get impaired by that. Yeah. Yeah, so that's where macular degeneration comes from, is from that degeneration of those cells in your retina. Okay, so big area representing foveal vision, the point with the best acuity, the best detail vision, somewhat smaller for your periphery, right? So the farther you get out in the periphery, the less detail you have, the fewer neurons you have devoted to representing that. Does that make sense? So everybody got the retinotopic map? Ooh, here's a fun fact. The retinotopic map is upside down and backwards. So why don't we see the world upside down? Does your brain flip it? Does it need to flip it? There's no need to. Have you ever experienced anything differently? No. And so your brain doesn't know any different. It's only from sort of our external point of view that things look upside down and backwards. To your brain, this is the way that it's always been. And so there's no need to actually do any flipping. But it's interesting because the lower part of the primary visual cortex represents the upper part of the visual field. And then the right hemisphere is going to represent the left visual field. So if you have damage, let's say let's put your damage right up here. So the upper part of V1 on the right side. You're going to have a scotoma, that was the word I couldn't think of on Monday, or a blind spot for what region of space? Left, upper, or lower? Lower, right. Yeah, so if the damage is in the right upper V1, then it's going to be contralateral and upside down. So it's going to be the left lower visual field, which I think is what this is, yeah. So if you have damage to all of V1 in one hemisphere, you're going to have a scotoma for the opposite side of space, where you're not going to be able to perceive what's going on there. If you have damage to, this is the lower left, then you'll have a scotoma here in the upper right. The size of the damage is going to be proportional to the size of your deficit. Why? Retinotopic map. Yes, exactly right. Because of the retinotopic map, right? It's a distorted map, but it is nonetheless a map, right? And so if you have a big lesion, you're going to have a big blind spot, a big scotoma. If you have a little lesion, you'll have a much smaller blind spot or scotoma.
So why does the brain have the part that's the most important, the fovea in the back, where it's most likely to be damaged?
We don't know. It's just the way that you developmentally, it's just the way that you develop, right? Or the way that your brain develops. Part of the reason why there's not, I mean, evolutionarily speaking, if you're going to have brain damage severe enough to cause this kind of scotoma, how likely are you going to be able to survive without medical care? Not very likely, right? And so if you're going to end up with this kind of brain damage in the era of evolutionary adaptivity, you're probably going to die. So there's no evolutionary advantage to like protecting this in the middle of the brain or something like that, because, you know, brain trauma kills without medical attention, right? It's only recently that we've been able to sort of treat these sort of things and observe this kind of scotoma happening, yeah.
This is really random. When I was a kid, they used to, my cousins, they used to say cross your eyes, and they would try to smack the back of your head. They said you'd stay cross-eyed. Does that have to do anything with that?
No, it doesn't actually work.
Okay.
Yeah, that might be a good paper topic, in fact. Yeah. No, because, I mean, I guess it could work if you damaged brainstem, because brainstem helps with like focusing your eyes and things like that. But if you're going to damage brainstem by hitting your cousin in the back of the head, I mean, that's you're hitting your cousin way too hard in that case. Right, I know. Just in general, don't hit your cousin in the back of the head.
Okay, I'm just feeling a little confused because Monday we talked about how the left eye, like the information for the left eye goes over to your left and the information for the right goes over to the right. So
Mostly, except for when it doesn't.
okay.
This is a good one. So half of the information crosses over. So it's not just the information from your left eye going to the right, it's half of the information from your left eye goes over to the right, half of the information stays on the other side. Yeah. Yeah, so it's the left visual field that's crossing over to the right, but that visual field information is coming from both eyes. Yeah, great question. I'm glad you asked that. Okay, everybody cool? So the retinotopic map does what? Yeah, it matches vision. So whatever is happening on your retina is represented in the primary visual cortex. And so, let's see, if we put it all together, do we have a slide putting it all together? Yes, here we go. So if you put it all together and if you take a look at everything that's going on in V1, this is sort of a representation of everything that's going on in V1. You have these neurons that respond to lines of a certain orientation, and the colors correspond to the various orientations. So red is horizontal, green is vertical, blue is sort of up to the left, yellow is up to the right. And then these stripes are ocular dominance columns, so responding to the right eye, the left eye, the right eye, the left eye. And then, oh no, where am I? There's another thing in here. Did I take it out? I thought I had another slide about blobs. Oh, let me show you blobs. There's an actual scientific no, not that. V1. Let's see. Yeah, that's okay. No, go back. I want like a big version of that. Okay, never mind. All right, so you have these hypercolumns, right, that represent all the different orientations of lines that respond to input coming from the right eye and the left eye. In the middle of these, you have areas that respond to color. And when you stain for these areas that respond to color, they look like blobs on the cortex. And so the actual scientific name for these are blobs. They're color blobs. So it's areas that respond to color in V1. So V1 responds to lines, edges, information coming from either or both eyes, and color. Anything else you need from the visual input? Maybe motion, but the complex cells respond to lines of a certain orientation moving in a certain direction. So you've got all of the information, it's all just kind of decomposed into its constituent parts. Does that make sense? Okay. Are you born like this? Is your brain ready to go, ready to see the world at birth? Or is there some development that happens? More experiments with cats. All right. Let's see what happens with the cat. They have 9 lives, so they have a lot of time to learn this. I think I'm going to skip forward a little bit.
By covering up the eye that has previously seen,
sorry,
soon causes the connections from the deprived eye to be re-established.
Okay, hold on. We're going to skip forward a little bit more.
This re-related by moving patterns. For example, on a television screen in front of the animal's eyes.
Okay, so you have an anesthetized cat.
Here is a deeply anesthetized cat, and you can hear the impulses from a single nerve cell picked up by a microelectrode implanted in the visual cortex. This cell is spontaneously active even when there is no pattern on the screen, but it discharges very vigorously if a vertical bar moves across the correct part of the visual field. This region is called the receptive field of the neuron. If the bar is horizontal, there is no response. Neurons in the visual cortex, then, are orientation detectors, and this one responds best to a moving vertical edge. The results from such experiments can be shown in a diagram. Each cell prefers a particular orientation. These lines represent all the orientations preferred by a sample of neurons from a single normal cat. We have conducted some experiments to see how far these orientation detecting properties can be influenced by the cat's early visual experience. In these experiments, we regulate the visual environment by keeping kittens in complete darkness from birth, bringing them out for periods of controlled visual experience inside special cylindrical chambers. Here, a kitten is being exposed to an environment consisting entirely of horizontal stripes, and it wears a ruff to prevent it seeing its own body. This particular kitten will be exposed to these stripes each day from the age of 3 weeks until it is 3 months old. All the rest of the time, it remains in complete darkness. What are the consequences?
They're well cared
of this
for.
restricted
They're okay.
visual experience? This is the same kitten at 3 months of age, seeing a normal environment for the first time. If we test its visual ability at this stage, we find that unlike a normal kitten of the same age, it has no visual placing reaction. It does not extend its paws to reach a surface below, although it does so as soon as it touches the table. And whereas a normal kitten backs away if an object is suddenly thrust towards it, this animal has no such startle reaction. It does not even follow a moving object, unless this makes a noise. These initial deficits seem to be failures of visuo-motor coordination, probably an effect of the kitten's lack of interaction with visible moving objects, rather than of its specific exposure to stripes. However, these gross problems don't last, and within a few hours, the kitten seems almost normal. Now, after several days of living in a normal environment, it has typical visual placing. It shows startle reactions. And it follows a moving object. But its vision is not really complete. Watch the way that it responds to a shaking horizontal stick. The kitten can see it. But if the stick is made vertical, the kitten no longer reacts to it. Remember that this kitten saw only horizontal stripes when it was young. If we record from the cortex in a cat like this, we find that there are no neurons that prefer vertical edges. The cells will only respond to those orientations which the kitten saw when it was very young. Presumably, this explains the animal's unresponsiveness to vertical contours. In the same way, a kitten reared in vertical stripes is found to have only vertical and near vertical detecting neurons in its visual cortex. This process of environmental modification of the cortex can also only occur during the same 3-week to 3-month sensitive period. In fact, we have found a period of peak sensitivity, about 28 days from birth, when only 1 hour's exposure to vertical stripes makes almost all cortical cells prefer vertical edges. So a kitten's early visual environment is crucially important for constructing the visual part of its brain.
I'm going to pause there. All right, so how much does this develop, this orientation selectivity, in cats? Like, are the cats born being able to see stripes of a certain orientation? No. They have to have some well-organized visual input in order to be able to do this, in order for their visual system to be able to set itself up. Same thing with the ocular dominance columns, the right eye versus left eye. Which makes sense, right? Because if you're born primed to receive input from both eyes. But if one eye doesn't work for whatever reason, the other eye can take over. And then after a couple of months, then that sort of critical window shuts down, and then it's much harder to change after that.
How does this work with, because like everybody's like right eye or left eye dominant,
Mm-hmm.
so how does that work with the hypercolor?
Yeah, good question. So right eye versus left eye dominance. So how do you know if you are right eye versus left eye dominant? So everybody do this and line it up with this little square right here. All right, and then bring it back to your eye.
The eye that you bring it to.
Yeah, so your dominant eye is going to be the one that you preferentially line up with. So I'm right eye dominant. How many of you are right eye dominant? Okay, you're probably mostly right-handed as well. Anybody left eye dominant? Okay, and that proportion is higher than the number of lefties that we have. We only have 2 lefties in here, if I remember correctly. Okay, so you're left eye down. Yeah, okay. Right, so if you have an astigmatism, if there's any sort of visual difficulty with one eye, you're going to favor the other eye, right? And so the cortical representation for that eye is going to be a little bit stronger. So that right eye versus left eye dominance, it's going to be more favored toward your dominant eye. And that can change. So we see sort of cortical plasticity that happens with people who lose vision or whose vision changes over their lifetime, things like that. But that's going to be much slower than during this critical learning period where only an hour's exposure can set up these orientation selective cells.
I've heard that you can be ambidextrously dominant.
You can be, yeah. Yeah, so those of you who are doing this right in the middle, yeah, you can be, and it can be situationally specific, right? So you might use one eye to look through a telescope or one eye for shooting or something like that, yeah. Unless you're crossed over, like if you're right-handed but left eye dominant, like shooting a gun becomes really
so
difficult.
annoying.
Yeah, yeah, yeah.
Do we know what the critical window is for humans?
Ooh, good question. Not specifically, right? Because ethics, right? So you can't do these kinds of experiments with babies. It seems to be probably very early on, within the first couple of, definitely within the first couple of months, probably the first couple of weeks. Newborns, you have kids. What do newborns like to look at? Faces, so newborns prefer faces, and you seem to be primed, so there seems to be a biological predisposition to look at faces. And the other thing that newborns seem to like is like high contrast things, right? So, you know, if you go to, let's see, Babies R Us is not a thing anymore, is it? They went bankrupt. Amazon, okay, so go to Amazon, look for baby products, and they're gonna have like bright colors and blah, blah, blah. And so, you know, you might want to buy, you might feel pressured to buy really expensive mobiles and things like that to give your kid lots of visual stimulation. I think Baby Einstein is still a thing, and they will be more than happy to take your money and sell you very complicated things. Kids don't care about any of that. Babies like to look at like the vertical blinds in the window, like those stupid shades that you all have in your apartments, or at least that we had in our first apartments. Like we, you know, spent some money on a nice mobile, and our kids didn't look at that at all. They just looked at the window. Because why? Lines, edges, nice high contrast, right? That's the sort of thing that they need for their visual input, and that's going to help set up their primary visual cortex. So as long as you get well-patterned input, your primary visual cortex will set up normally. It's only under these very special circumstances where you keep the animal in complete darkness and only expose it to horizontal or vertical stripes that you get things that are weird, right? And so that's why we know we can do this with cats because of ethics, but we can't do it with humans. Okay. Any other questions about V1? Are we on time? We've got a couple of minutes. Yes? Yeah, so everybody's retinotopic map is going to be slightly different depending on your very early visual input. Yeah, which is not to say that, you know, some people are going to have better representations than others. It's just going to be different representations. Yeah. So being able to map this from one individual to the next is going to be very difficult. Yeah.
Is that why cultures tend to differ in what they prefer to see in architecture?
Oh, yes, there are some theories about that. Like we like right angles and things like that, and if you are like exposed to that early on, you may prefer that. But if you're exposed to a more organic kind of architecture with like soft edges and round walls and things like that, you may prefer that later on and actually have difficulty perceiving right angles. And so there are some ethnographic experiments on that sort of thing. Yeah, interesting. Okay, so this is a schematic of the visual system, and we are on step 3 after 2 days. We need to hurry up. Okay, very broadly. So once you get out of V1, you see this really interesting divergence of the visual input. So V1 cares about what? Lines and edges. But you can see more than lines and edges, right? You can see complex objects. So how do you get complex objects from basic lines and edges? Yeah, so just like you go from seeing individual points of light to seeing lines by combining and stacking up the individual points of light, now if you want to see angles, for example, how would you get an angle out of two lines? Just put the two receptive fields together and have those two cells converge on a, let's go back, on a V2 neuron. So V2, not surprisingly, cares about angles, more complicated lines and edges, basically. V4 cares about some pretty basic objects. And then once you get into here, into inferior temporal cortex, this part of the brain cares about objects. So you go from very basic sort of line sketches to more complicated objects and faces, and Jennifer Aniston is somewhere in here. Yeah, the cells that they recorded, the Jennifer Aniston cells, were in kind of this area. Okay, so that's one stream, and that's called the ventral visual processing stream, or the what stream. So these areas in inferior temporal cortex care about the objects that are out there in space. They'll respond to their preferred stimulus, which are going to be objects. Okay, you've also got another processing stream that goes dorsally up here into the parietal lobe. Does anybody know what the parietal lobe cares about? Not objects. Spatial, yeah. So this is your where processing stream. So you've got the what and the where processing streams. So the where processing stream cares where objects are out there in space. And remember, primary somatosensory cortex is up here. So it's going to combine knowing where your body is in space with knowing where objects are in space that will allow you to do things like reach out and pick up the water bottle, right? It'll guide your visual actions.
So is this why we see lines? Because then it can be differentiated between both areas of the brain?
Yep, yeah. So lines and edges, you're going to need lines and edges for both of those things, right? So identifying what objects are out there, right? But also identifying where those objects are. Yeah.
The stream that goes into the parietal lobe is the dorsal.
Correct. Yep. So you've got the dorsal stream, which is the where stream, and the ventral stream, which is the what stream. So dorsal and ventral. Okay, so one last point and then I'll let you go. What if you have damage to the ventral stream? What kind of impairments would you expect? Yeah, you would have what are called object agnosias, where if I hold this up and ask you, what is that? I don't know. But if I ask you to reach out and grab it, you'd be perfectly able to do it. And then once you have it in your hand, you could identify it based on the shape. So, which is crazy. If you have damage up here, what kind of deficits would you expect? Right, exactly. So if I hold this up and say, what is it? It's a marker. If I ask you to reach out and grab it, that sort of thing, right? I'll show you some videos next time of people who have these specific types of damage. It's really, really interesting. Okay, thanks everybody.